I would like to invite Vijayalakshmi ma'am.
Good evening, everybody. My talk is not going to be on gene therapy, but it's going to be on one of the systems that will be definitely essential for basic science research to understand and delve into gene therapy. Today, I'm going to speak on the importance of in vitro systems in understanding the brain functions under normal and diseased conditions. This is not working? Okay. Yeah. Okay. So, as we all know, we have for basic science, we have a lot of animal models. So the question is, why do we need in vitro systems when we have so many animal models with which we can study several neurological and the brain disorders? So, next please. It's not working. So in the first place, what is an in vitro system? An in vitro system is something that we grow in vitro, that is in glass, outside the biological system. So any organism or cells or biological components which we grow outside its normal biological context forms the in vitro system. For example, the cell culture system. One of the most important form of cell culture system is the primary cell culture system, where we can directly take the tissue from the animal, say for example, brain tissue of our interest, and then dissociate the tissue and grow that to get the primary cultures, or depending on the need, we can just take the explant and grow it outside, which will form the explant culture. While we do that, there are several procedures. Due to the want of time, I will not get into these details now. So how do we do this? When we have to grow it outside, it can be prone to contamination by other microorganisms. So we have to take a lot of sterilization precautionary measures, and we have to have dedicated spaces like incubators for maintaining these without getting contaminated, and also we have to keep on monitoring these cultures whenever upon the experiments. So if we just go to the brief history of this cell culture system, it was by Ross Harrison who first discovered that cells can be grown outside the body. So at that point of time, tissue culture was just a curiosity, but it is now considered as one of the medicine's 10 greatest discoveries is about this cell culture system. After Ross Harrison, there have been many other methodological changes to improvise the cell culture conditions, and these people, further scientists, they resolved the issues that were there to culture the primary cultures or to grow the cells outside by providing better culture dishes, growth media, and also to control the death of the cultures. So we have various cell culture media, that is specialized medium, that the nutrients through which we give the nutrients to the cultures. So we have different media such as natural media or chemical media. Next slide, please. And in the natural media, we have plasma or serum from animal sources, which will support the growth of these cells which are grown outside. Can you please go to the next one? In chemically defined medium, these are known components. These are usually not from the biological sources. Next slide, please. For example, here we have the composition of one of the media which will have all the macronutrients and the micronutrients that is essential for the survival of the cells outside its natural source. Next slide. I'll not get into the technical details again for the want of the time. Can you please go to the next one? So what are the different applications of the cell culture system? Ranging from right development of the monoclonal antibodies to vaccine production, and also various applications are there where we can even have disease models at the cellular level. So because of its vast applications, and cell culture systems have a lot of advantages. Next slide, please. So in my today's talk, briefly, I will be focusing on in vitro systems in research on neurotoxicology and translational neuroscience. So here you have 4 very prominent people. There is one common thing in them. Can anybody tell me that? What is that common thing? all of these suffered from a neurodegenerative disease called Parkinson's disease. Okay, so what is this disease? It is a movement disorder mainly affected by the adults or in old age or above 40 to 50 years of age. Because of the loss of specific neurons in one of the specific brain area, namely the dopaminergic neurons in the substantia nigra area of the brain. Can you please go? So what happens is there are several toxic molecules which we might be exposed to, say for example the pesticides, etc. One amongst the toxic molecule which can induce Parkinson's disease is called MPTP. This toxin, through blood-brain barrier, it enters into the nervous system, and there the helping cells of the neurons, that is the glial cells, first take up this MPTP and then convert it to a form which MPP plus, which binds to the neuronal, that is the dopaminergic neurons through the dopamine transporter. Only when it is converted from MPTP to MPP plus and the dopaminergic neurons which have the transporters on them can bind to it and take it in. Once it goes in, it causes several neurotoxic changes and that leads to the neurodegenerative disorder. So all these understandings were specifically brought about by detailed research on the in vitro systems. Can we go to the next one? So the other neurotoxins which can cause Parkinson's disease is the pesticides such as paraquat, maneb, etc. and even the metals such as manganese, etc. Can you please go? So we have endogenous toxins and exogenous toxins. And endogenous, whenever the normal nitric oxide or dopamine levels are exceedingly higher within our cells, then also it can lead to the disorder. So how does in vitro system help us to investigate this? These neurotoxins not only cause the neurological disorders such as Parkinson's disease, which is a movement disorder, but also it can cause psychiatric disorders. So cell culture system plays a major role in helping us to understand. So one of my senior colleagues always says, if the genes load the gun, it's the environment which pulls the trigger. So the toxins presence within the environment will cause this neurodegenerative disorder. Can you please go to the next one? So again, I will talk about how the cell culture system or the in vitro system helps in understanding this neurological disorders. Oh, that was too early. Then I think, okay, quick. So we have cell culture system and animal models also, wherein we can have genetic models by knocking in the genes or gene knockout studies in these cell culture systems to understand the detailed pathways and the mechanisms by which we can get the details of how exactly the disease occurs because of the genetic changes. I will not get into all these because of the want of time. Can we just go to the next one, please? Please, the next one. Yeah, we have different cell culture systems, cell lines and primary cultures. Next. So depending on the need, we can decide whether it is cell line or primary cultures. Can you please go to the... Or we can also co-culture them. Two different cell types we can co-culture and then understand the disorder. Or we can even have stem cells or patient-derived cells. We can take nowadays the technology has advanced so much that even from hair follicles or skin, blood, and also even from urine samples, cells can be extracted and it can be reprogrammed to become patient-specific cells, those which can again be differentiated into neurons, glia, or any cell type and can be studied. We will just go. I will skip a part of this. Can you please go further? Yeah. I will, next, please. Next. No, I will move this because I thought we have more time. Because of the time constraint, we have to, yeah. So, one of the application is to understand, another application is to study the in vitro studies on the translational research. So, next one, please. So what do we mean by translational research? Any basic research which can help in development of new drugs or therapeutic modalities or even biomarkers for proper diagnosis of disease can be facilitated through translational research, and in vitro system plays a major role. Can we go to the next one? So here we have the famous ice bucket challenge, and this was majorly raised for, the funds were raised through ice bucket challenge for one of the neurodegenerative disorder, namely amyotrophic lateral sclerosis, which even Stephen Hawking suffered from. Can we go to the next one? So it is a motor neuron degenerative disorder where the motor neurons, that is the neurons which bring about the movement, they are getting degenerated or they die off. And similarly here also, this is an adult onset disorder. And within 2 to 5 years, most of the patients die once they get this disease. We do not know the cause of the disease. 5 to 10% are familial, but the remaining 90% are sporadic. In our lab, we focus on understanding the sporadic form of the disease, so for which we use the CSF samples from the patients and we add it to the cell culture system. So we derive cultures, that is of rat or mouse or even human embryonic stem cells. We grow them in dish and we add the CSF from patients to these cultures and we study how this disease occurs. Can you please play? In this animal model, the video. If you can, it's not happening. If you can see this video, I hope it plays. No? Yeah. So here, in this experiment, the third rat on the rotating wheel is injected with CSF from ALS patient. When the speed of this rotating wheel is increasing, we can see that the rat falls off. That is because the CSF has some toxic factors in it, which you can see that the animal falls off. Unlike the other normal animals, you can go to the next one. So because of the presence of some toxic factor, so in the cell culture system, we can see that on the left panel, under normal conditions, the neurons are normal, but when we add ALS CSF, we can see that the neurons are dying. The black arrows will show that there is bleeding of neurites, the cytoplasm is affected, that is the cultures, the neurons are dying. Go to the next one. So similarly, in other cell line also, we found that motor neurons, the neurons will die off, and they also undergo apoptotic changes. Next, please. So there is apoptotic mode of cell death, which is happening upon exposure to ALS CSF. Next one, please. Next. And what we see is, in addition to this, wherever the most important proteins of the neurons are essential for its functioning, it gets aggregated and ubiquitinated, and they cause death of the motor neurons. Next, please. So on the right panel, you can see down, the mitochondria are also affected because we can see that mitochondria are bulged, or we can see the presence of apoptotic bodies by electron microscopy. And what we see is here on the helping cells, the glia are helping cells for the neurons. On the left-hand side, what we see is the flat astrocytes, which means these are the type of the cells which will protect the neurons. But the friends turn into four, that means they become a toxic form and take the fibrous form, where they, instead of protecting by releasing growth factors and supportive factors, they start killing the neurons by releasing more toxic factors. Next, please. So from all this study, what we understood is, there are different cell types like astrocytes, microglia, muscle, all these are affected, be it in animal model or in vitro conditions, to say that the CSF has something in it, CSF of ALS patients has something in it to kill the neurons or bring in the degenerative changes. Now the next most important question is, what is it in ALS CSF which is causing this degenerative changes? So, next please. When we did mass spectroscopic analysis, we found that among more than 32 proteins which were upregulated, there is chitotriosidase, an enzyme which is upregulated by more than 20 folds. Can we please go to the next one? And by ELISA also we found that it is upregulated by more than 20 folds. Next please. Yes, please. So once we have identified this toxic factor, we have now known that worldwide people have taken this protein, the study by this protein ahead, and they've shown that it can be a putative biomarker. Therefore, study on in vitro conditions will facilitate us to take it to the translational level. Further research is going on, and I will not get into the details because of the want of the time. Thank you.